Hello everyone, welcome back to exam prep, and for this video we are going to do the May June 2021 exams paper 2 variant 1 as promised. So if you watched the previous 2021 May June exam video, I mentioned that I will do the exam papers 1, 2, 4 and 5 focusing only on variant 1, and just use that as a kind of revision for the students who are preparing for the October-November exams, so that you can sort of have an idea of what to expect and how to answer questions properly. So this is the full paper 2 exam. For those of you who are writing, you should know that paper 2 is a 75-minute paper. That means it's 1 hour 15 minutes. Just like paper 1, it only focuses on chapters 1 to 11. So you don't need to study the chapters from 12 to 19 for paper 2. Same way you don't need to study them for paper 1. It's the second paper for those who are writing only at AS level. And if you're writing at A level, you definitely know that you write all the 5 papers. So let's get into it. Hopefully I can give you some tips on how to answer some of these questions, and hopefully those who wrote the exam, I don't think they would care at this point, but in case you just want to revise and see what you did wrong, then this is a good opportunity to check it out. All right, let's start with the first one. So the first question here says figure 1.1 is a transmission electron micrograph of cells from duckweed, Spirodella oligorhiza. I haven't done microbiology in such a long time that I just forget how to pronounce these names. Anyway, let's look at the first question. It says calculate the actual width of the cell labeled X. Write down the formula you will use to make your calculation. Show your working and give your answer in micrometers to 1 decimal place. So whenever you have a question like this, I tell students to pay very close attention to the instructions and not to jump. So first of all, it says you need to write down your formula. So this is the space for your formula over here. And then show your working. So you have to show your working and give your answer to 1 decimal place. This means that if you write your answer, because sometimes students are not convinced, right? So say your answer is maybe 4.07. Students tend to feel the need, I don't think that's a good example. Let's say your answer is 4.04 rather. 4.04, and obviously you can't approximate 4 to 1, so students tend to be unconvinced in writing 4.0 as the final answer, even though it says to 1 decimal place, because they for some reason feel the compulsive need to show that there is a number after 0. Please do not do that if you're in a situation like this. Always stick to what the question has asked you, which is to give your answer in micrometers to 1 decimal place. So since we are calculating the actual length of the cell, so in this case you already know that magnification is equal to image length over actual length, right? And obviously if you're going to calculate the actual length, you do some cross multiplication here, and then it's going to be actual length is equal to image length divided by magnification. You've been given the magnification here as times 4275. I don't feel this is inaccurate because I sniped this image, so I'm not sure if the length would be accurate if I were to measure it. So what you do here in this case is that you take your ruler and you calculate, you measure rather the length of this line. So you can see where it says X, you measure from there to there. Once you've done that measurement, your answer should probably be in millimeters or centimeters. Okay, then you calculate what this would be in micrometers, because remember your instruction says to give your answer in micrometers. Then you convert millimeters to micrometers, or you go from centimeters to micrometers. So from centimeters, I like to do this step by step so that students don't get confused. If you're going from centimeters to millimeters, you multiply by 10. Okay, if you're going from millimeters to micrometers, you multiply by 1000. Okay, so that would give you your value in micrometers, and then you just take that value, because this would be your image length or image size, and then you just take that and divide it by the magnification, which is usually given at the bottom here, and then you write your answer, whatever that may be. All right. So, and make sure your answer again is to 1 decimal place. If you write more than 1 decimal place, even if everything you have done is correct, your answer will be marked wrong. Okay, let's look at the next question. Here it says table 1.1 lists some biological molecules found in plant cells. Complete table 1.1 by choosing one letter from figure 1.1 that indicates a cell structure where each biological molecule is found. So this is what you have to look at, and you've already been told that these are plant cells. Again, please, whenever you are answering questions like this, always pay attention to the instructions or the information that's been given to you. And if possible, if you have your pencil with you, take your pencil and underline the things that are important so that when you're answering the question, you can keep those in mind. So where would you find DNA? If this is a plant cell, where would we find DNA? Well, DNA typically would be found in the chloroplast. You can find DNA in the chloroplast. You can find DNA in the mitochondria. And again, if you've watched the paper 1 video, you know about the endosymbiont theory, so this should not be surprising to you. And you can also find DNA inside the nucleus. All right, so in this case, when you look at this, you can see that B seems to be very much the nucleus. That's where your DNA would be concentrated, and so I would go with B, but you can actually go with C as well. C looks like the chloroplast, or you could even go with A, because I think A might be maybe like I think B is the nucleolus and A is the nucleus. I'm not entirely sure about that, but very convinced that B would be a correct answer. Where would you find cellulose? Cellulose you would find in the cell wall of the plant. So remember, you don't find cellulose in the cell membrane, it's in the cell wall. It gives the plant its rigid structure. So you simply need to look out for the borders of the plant. So if you look at E looks like a plant border. So it's like where one cell ends and another one begins. So I would go with E over here. In phospholipids, where would you find a phospholipid? The phospholipid would certainly be around a membrane, or in the membrane of the cell, or it could be even the membrane of the chloroplast itself. So in this case, you can actually go with either A or you could go with C, but I would just go with A here. And where are you likely to find histone proteins? Always remember that histone proteins are associated with the DNA, and so you would find histone proteins where your DNA is, and that would be B. Okay. Name a cell structure visible in figure 1.1 where ATP is synthesized. So what is visible here that we can see? I know it's not ideal because this is quite a small image, but we can see the chloroplast. I mean, those are the chloroplasts quite clearly. So in this case, I would go with C, or it says state the name, so it doesn't say state the letter. And again, this points to the importance of you reading the instructions, because sometimes students would say, well, if I was doing letters before, I can just continue with letters since it's the same question B. Please always read the instructions. It says states the name of a cell structure where ATP is synthesized, and in this case, it would be the chloroplast. Okay. And then the last one on this slide says, name a cell structure that produces mRNA. For you to produce mRNA, you need to get RNA polymerase to go into the nucleus where the DNA is in order to copy the DNA. So I always say to students here, in this case, RNA polymerase is not a cell structure, it is an enzyme. In this case, the cell structure that would produce mRNA would be the nucleus. Okay, it's also not DNA. DNA can't be the answer. DNA is a biological molecule. It is not a cell structure. Describe the evidence from figure 1.1 that shows that the image is a transmission electron micrograph. So usually these kinds of questions usually come on and they're just 2 marks. But what you can tell is, for example, you can tell that transmission electron micrographs, by the way, give higher or better detail than you would find on a light microscope, obviously. So something you can state here is that you can see the chloroplast, the inner structures of the chloroplast more clearly, because if you look at these chloroplasts, you can see what is called the grana. So in case you're wondering what that is, please watch the videos on photosynthesis where I explain what the grana and the thylakoids are. You can also see that the magnification is quite high, and the resolution seems to be a lot better. So I think 2 things here that you can say, because this is for 2 marks, is that there is a high magnification of the image and you can see more detailed information such as the grana in the chloroplast, and that should give you 4 marks. Okay, let's go to question number 2, and just something I should have said at the beginning. When you're doing paper 2, you have 6 questions and therefore 60 marks, and you have 1 hour 15 minutes to get through it. And obviously everything is structured questions, so just putting that out there so that you are aware of how many questions you have to get through, and you have to answer all questions. Oh well, that's the instruction. Except you run out of time, you should try to answer all questions. All right, let's look at this one. It says describe the hydrogen bonding that occurs between water molecules in figure 2.1. So over here, please pay attention to the verb. It says describe, all right, the hydrogen bonding, which means that in this case, you are not allowed to just come in and say water has strong forces of cohesion, water has strong forces of adhesion. That's not what this is asking you to do. It is asking you to look at this image over here and say what you can find, or more or less use your understanding of the bonds between water molecules to answer the question. So first things first that you would see are these dotted lines, or first things that I see are these dotted lines between the water molecules. So this is one water molecule over here. Actually, I should erase this big blankety thing so that it doesn't interfere. So that's one water molecule over there. This is another water molecule over there. Can you see that? And that's obviously another water molecule. And between those water molecules, you will see these dotted lines. Can you see those? Those are forces of attraction, all right? So in this case, you would be able to say that there are forces of attraction between the oxygen molecule of one water molecule and the hydrogen molecule of another water molecule. Then something else that you can say is that hydrogen bonds are weak bonds. That's basically a standard thing, that they are weak bonds. And something else that you can also mention is based on the size, and I know this might be reaching for some of you, but based on the size that you can see for each atom, look there, oxygen seems to be a bit bigger than hydrogen atoms that are in bonds with it. And so what you can say in this case is that the electrons seem to not be shared equally between the two, and as a result of that, but that's between oxygen and hydrogen, and as a result of that, water is a dipolar molecule. When we say water is dipolar, what that means is that oxygen still carries some some negative charge, while hydrogen carries some positive charge. For those of you who do chemistry, I'm sure that this is just pretty standard information. For those of you who don't, please pause, rewind, listen again, so that it's a bit clearer to you. And if you have questions, just post them in the comments. Okay, let's look at the next question. Looks like enzymes. I love enzymes a lot, so let's see. The human enzyme salivary amylase is composed of one polypeptide. Figure 2.2 represents the structure of a molecule of salivary amylase. And then just to give you some background, salivary amylase is the enzyme that's in your saliva, and it starts the breakdown of starch the moment you start to eat. So even before the starch gets to your stomach, the enzyme starts to break it down already. And then here it says, question 1, explain the role of hydrogen bonding in maintaining the secondary structure of proteins such as salivary amylase. So the role of hydrogen bonding in maintaining the secondary structure of proteins, here pay attention to what it is asking you. It is saying the secondary structure. You know that the secondary structure of proteins, if you have watched the videos I made on proteins, are the alpha helices, or for singular you can just say alpha helix, or the beta pleated sheets, or you can just say beta sheets. And please take note of how I write alpha helix and beta. Please don't go to the exam if you're ever asked to write it and just write a helix. That will be marked wrong. You're better off, if you don't know how to do the sign, writing the full word alpha. Okay, just pointing that out. Let's get back to the question. So what is the role of hydrogen bonding in maintaining the secondary structure of proteins? In this case, you always need to remember that the hydrogen bonds are what maintain the alpha helices and the beta pleated sheets. So please, if you don't know that, watch the videos I made on chapter 2, biological molecules. Just go to the playlist function, select chapter 2, and all the videos are there. I think there's a separate video for proteins, so that would be very helpful for you. So in this case, it would just be they maintain the structure of the alpha helices and the beta sheets. Then it goes on to ask again, it says, explain the role of hydrogen bonding in maintaining the tertiary structure, of proteins such as salivary amylase. So when you're looking at the tertiary structure, you're dealing with 4 different levels. you're dealing with 4 different types of bonds rather. You're dealing with the ionic bonds, you're dealing with the hydrogen bonds, you're dealing with the hydrophobic interactions. I'm not going to write all of them out. You're also dealing with disulfide bonds. So what do hydrogen bonds help to do? Hydrogen bonds help with further folding, so they improve or they help with the folding of the protein into its 3D shape. Something else that they do is that they help with the formation of the active site of the enzyme. So that's something that you would bear in mind to answer this question. And oh yeah, you only have to give 2 points because it's just 2 marks and you should be fine. C. Outline the importance of water as a solvent in plants. Obviously, you know that water is a universal solvent, and what water does is that, for example, within the human body, it would transport solutes. It does the same thing in plants. If you have not watched Transport in Plants, please do that on chapter 7, and you would learn that water it transports things like your mineral ions through the xylem, but even in the phloem, water is needed to transport things like sucrose. So that is very important. Water also helps with the occurrence of metabolic reactions. So even within the human body, I know that this is referring to plants, but just pointing out that even within the human body, almost everything that occurs in our body occurs in the space of water. So there is no dry spot in your body where some reaction is happening on its own in a corner. Something else that you can say about water is that in plants you have what we call the vacuole. The vacuole is this large sac, and you might see it when you look at a detailed diagram of a plant cell. And in the vacuole, it's like a storage thing as well, and water is used for that storage of solutes. All right, let's go on. Okay, question 3. I think this video would be much shorter than the other one we did on paper 1. That was quite a long video. All right, now let's look at this. Vieskin tubing can be used to investigate the properties of cell membranes. So wherever you see Vieskin tubing, please know that you're most likely going to be dealing with something that has to do with osmosis. Okay, so Transport in Plants, not Transport in Plants, Cell Membranes and Transport, chapter 4. Please go watch those videos if you haven't. Trust me, these are very helpful. A student carried out an experiment to investigate osmosis using Vieskin tubing. And just by the way, if you are not a home registered student, or you go to a school, please ask your teacher if they haven't done this with you to please demonstrate it so that you're able to see it play out, because I really feel like when students do the Vieskin tubing in action, it helps them a lot to understand. So an outline of the student's investigation is shown below. So over here we have a ruler, you have the Vieskin tube, there's the Vieskin tube, you have the meniscus, and obviously you have a sucrose solution here, and then top of the north of the Vieskin tubing. And over here you can see 15 centimeter cube of sucrose solution. And yeah, well, this is just an outline of what is happening here. So what you can see here is that this is our Vieskin tubing. This over here, I'm just going to circle it. Hope I can do that properly. There. Okay, and then it's placed into another tube, and you can see that that other tube also has its own what's it called, is its own liquid in it. So this over here has sucrose. Let me just erase that line so that you can see. So there's sucrose on the outside here, okay, and there's some on the inside here, and obviously you use the ruler to measure whatever changes in the levels of liquid that you can see. But let's read some more. 6 pieces of Vieskin tubing were filled with 10 centimeter cubes or cubic centimeters of different concentrations of sucrose, and you can see there's 0, 0.4, 0.8, 1.2, 1.6, and 2.0. The height of the meniscus of each solution in the Vieskin tubing was measured. The pieces of Vieskin tubing were put into test tubes containing 15 centimeter cubes or 15 cubic centimeters, whichever way you pronounce that measurement, of 0.9 mole per dm cube sucrose solution. After 20 minutes, the pieces of Vieskin tubing were removed from the test tubes, and the height of the meniscus in each was measured. So again, just to point out what that is saying to you is that you start off with this. This is your Viscount tubing over here. You put in different concentrations. As you can see, sometimes 0 means there is nothing there, just water. So just water, or you could put in 0.4, 0.8, whatever. And then you take that Viscount tube and you put it into a test tube. And in that test tube, you have 0.9 mole per dm cube of sucrose. Okay. And after 20 minutes, you then measure. If you measure what has happened, let's just put it that way, between the liquid on the outside, that's the sucrose on the outside, and the sucrose on the inside of the Viscount tube. So the results are shown in 3.1. So these are the results from that experiment. So you can see there, I've just put this here, the drawing just again so that you can remember what the experiment looked like. So over here where we had no concentration of sucrose inside the Viscount tubing, the height, difference in height of meniscus after 20 minutes was minus 12. That means that the height reduced. Okay, and I'll explain this to you just now. 0.4 the height reduced, this the height reduced, blah blah blah, and it goes on. So let me explain exactly what has happened here. You started off with the Viscount tube, and I'm going to draw my own over here, even though I can't draw. You started off with the Viscount tube, and in this Viscount tube you put in let's see, this was 0.0. okay, that means it's just water. And then you took that Viscount tube and you put it, I think I should use a different color so that it's a bit clearer. I'm going to use a blue color. You took that Viscount tube and you put it in the test tube, and in that test tube you put sucrose of 0.9 mole per dm cube. This is 0.0 mole per dm cube, so it's just water over here. Now if you're thinking of this, considering that you're investigating osmosis, the first thing I will ask is, what is the definition of osmosis? Osmosis is the movement of water from a region of high water concentration to a region of low water concentration through a semi-permeable membrane. The semi-permeable membrane in this case is the Viscount tubing. All right, so now in this case we have water here, and here we have 0.9 mole per dm cube of sucrose. What's going to happen in this case is that water will move out of the Viscount tube into the sucrose solution, simply because the sucrose solution has less water potential than the water in the Viscount tube, because the water in the Viscount tube doesn't have any sucrose in it. So as a result of that, because the concentration of sucrose is higher here, water will move in naturally. And that then explains why when you look at the results, the length of the water, or the height of the meniscus in the Viscount tube, decreases by 12 millimeters. Does this make sense? I'm speaking as though I can see you guys. I know I can't, but I'm just thinking, I'm imagining myself in the classroom with my students at this point, who, by the way, are not my students anymore because they have a new teacher. But I hope that makes it clear. So in this case, when we have, let's draw that again. So this is our test tube, and again we have 0.9 mole per dm cube in here, and then we have our Viscount tube, and in our Viscount tube we had 0.4 mole per dm cube. Again, the concentration of sucrose in the Viscount tube is less than that of the concentration of sucrose in the test tube. That then means that the water potential in the Viscount tube is higher than the water potential in the test tube. So water will move out, and that explains why you still have a decrease in the length of the meniscus in the Viscount tube. And the same thing goes on. But the moment you increase the concentration past what is in the test tube, water will move out from the test tube into the Viscount tube, and so that's why you start to see an increase in height instead. So I hope that explains it. So I think I've just answered this question. The Viscount tubing used by the student was not permeable to sucrose. So they're telling you this because they want you to think in terms of water. So many students, again, can make the mistake of saying sucrose is the one that's diffusing. But when you're investigating osmosis, you're investigating the movement of water and not the movement of a solute. All right. So in this regard, The Viscount tubing was not permeable to sucrose, that makes sense, but it is permeable to water. So that means that in this case, to answer this question for 3 marks, you have to show that the concentrations, they say explain the results. So you can say that when the concentrations in the Viscount tube were less than that of the concentration of sucrose in the test tube, water moved out of the Viscount tube because of osmosis. So osmosis is the net movement of water. Water moves down its concentration gradient, and when the concentrations were higher in the Viscount tube, water moved into the Viscount tube, resulting in the height of the meniscus. And just explain it in terms of osmosis, but you only have to give 3 different points for you to get 3 marks here. I hope that helps you. When red blood cells are placed in water, they are destroyed by bursting. So in this case, I just want to point out that that term is called hemolysis. All right, so when you place red blood cells in water, I don't know if this is important for the question, by the way, I haven't looked at the question, but just saying that whenever I see questions like this, I tend to first of all ask myself, well, what does this term mean? So that if it comes up later on, I am able to recall it, because sometimes you remember some things in the exams, and then when you're about to write them down, for some reason, they just fly out of your head. So always make notes of what you're writing. So when red blood cells are placed in water, they are destroyed by bursting. The student also investigated how red blood cells are affected by immersion in solutions of sodium chloride of different concentration. Blood samples of the same volume were added to solutions of sodium chloride in separate test tubes. After 10 minutes, the students took 0.1 cubic centimeter of the blood samples from the test tube and estimated the percentage of red blood cells that had burst in each blood sample. And these were the results. Okay, so concentration of sodium chloride, you can see that at 0, when there's no sodium chloride at all, it seemed like 100% of the cells burst. Okay, and even at 0.05, which is a very low concentration, 100% of the cells burst. So percentage of the red blood cells destroyed by bursting, and I'll explain why they burst to you just now. And you can see over there, oh, I think this is about 0.025, and then that's 0.05. But as you see, as the concentration of sodium chloride increases, the percentage of red blood cells that are bursting starts to decrease, and by the time you get to 0.15, the concentration of the percentage of red blood cells destroyed by bursting is 0. Now, why do red blood cells burst when you put them in water? You need to remember that you are dealing with what we call an animal cell. So red blood cells are animal cells. All right, they have a cell membrane, but they don't have a cell wall. So it means that there is nothing that gives them that rigidity and support that plant cells might have. And if you remember correctly, cellulose in the plant cell wall gives rigidity and structure, and also prevents the cell from bursting, because even if the cell membrane were to expand, it would meet with the cell wall, which then sort of keeps it within that space. Animal cells don't have that. So it means that if you take an animal cell and you put it in a solution of water, again, still explaining osmosis, the animal cell has solutes in it. Okay, it has all these organelles, it has things that it has stored in there. So many things are happening within an animal cell. When you place it in a solution that contains water, water will move into the cell. All right, because the water potential on the outside is higher than water potential on the inside, so water will move into the cell. Now, because there is no cell wall, the cell will start to swell because it's getting a lot of water, much more than it requires. And if it gets a lot of water to a certain extent, it will burst, and that bursting is what we call hemolysis for red blood cells. So let's see what the question says, just giving you that explanation ahead of time. Describe and explain the effects on red blood cells of immersion in different concentrations of sodium chloride as shown in figure 3.2. So again, bear in mind that the verb here says describe and explain. Okay, so describe and is in detail. What that means is that you can look at the graph, and I would say for you, I would say just give descriptions, maybe 2 descriptions, and then give 2 explanations that match those descriptions. So 2 descriptions would be that at low concentrations of sodium chloride between 0.0 and 0.05, 100% of the red blood cells were destroyed by bursting. Then you can explain that by saying this is due to the fact that water will move in, because 0.0 to 0.05 means that the water potential of this solution is still high. So water moves into the cell, causing the cell to swell and burst. Okay, so increase in cell volume, internal pressure, all of those kinds of things. That would be the explanation for that. Then you can go on and say between, let's see, I think this is 0.014 or 0.14, sorry, and 0.25, there is no bursting of red blood cells. And what this means is that you can say that the water potential between the cells must have been the same, and as a result of that, there was no net movement of water. So that means that the water does not, so that means that the cell does not take in water. Or you could also say that the water potential of the cell might be higher. All right, so it's possible that at this concentration of sodium chloride, the water potential of the cell is actually higher. So the cell is not bursting, rather it is losing water to the surrounding environment, which might mean that it's shrinking. So you don't have any clear explanations for that, but that is how you do describe and explain. Describe, you look at the graph and you spot a trend and you report it, and then you give an explanation for that trend, and then you look at again what is another trend, and you can also choose this trend over here, by the way. All right, you can say that as the concentration of sodium chloride increases in the solution, percentage of burst red blood cells decreases, and you can then explain that the water potential difference is not that high, and so no internal pressure is created. But the point is, always take the trend, look at it, and then give an explanation for why you think it's happening. That's how you answer, describe, and explain successfully. Please don't only describe. If the question says describe and explain, you will not get full marks. Let's look at question 4. We're making good progress because this paper, like I said, is only 6 marks. The induced fit hypothesis and the lock and key hypothesis are used to describe the mode of action of enzymes. Explain the induced fit hypothesis. So here you have 3 marks for this, which again means that you have to give 3 points. So in this case, in the induced fit, what you should remember is that the substrate does not fit perfectly into the active site of the enzyme. As a result of that, the enzyme's active site will change shape or modify its shape in order to fit the substrate. And then, due to that change or that modification, the substrate is able to bind to the site to form the enzyme substrate complex. And if you write those, you will get your 3 marks. Okay, question B. Radish plants contain the enzyme peroxidase that catalyzes the breakdown of hydrogen peroxide. Students investigated the effect of increasing the concentration of hydrogen peroxide on the activity of peroxidase extracted from radish. The results of the investigation are shown in figure 4.1. So this is figure 4.1. Again, what you can do here, or what is coming to me as I read the question, is I'm already saying to myself the enzyme is peroxidase and the substrate is hydrogen peroxide. So this is S and that's E. And now they investigated the effect of increasing substrate concentration. I've deliberately put it this way because when you learn enzymes in class, those are the things that you learn. What are the effects of increasing substrate concentration? What are the effects of increasing enzyme concentration? So putting it in the context of how you are taught in the classroom would help you easily deduce what you need to answer here, or how you should answer. All right, so always do that. Even if it doesn't come up as something you need in the answer, it's always good with your it's good to help you think about how an answer should be structured in any case. So let's see here. So these are the results, and if we look at this graph, the rate of the reaction, obviously when the substrate concentration is 0, so is the rate of the reaction, because the enzyme has nothing to act on. But as you can see, as the substrate concentration increases, the rate of reaction also increases until it gets to a certain point where it flattens off. And this can be explained by the fact that it gets to a point where as you increase enzyme concentration, as you increase substrate concentration rather, the enzymes become saturated, and as a result of that, the rate of the reaction is stalled. And I think I explained this on the video with enzymes or on another video, but I will see what the question is asking before I start to go on a tangent. Okay, so the question says, explain the effect of increasing the concentration of hydrogen peroxide on the initial rate of reaction as shown in figure 4.1, and this is for 3 marks. Okay, so first things first that you need to say is that in this case, between let me see, between 0.0 mole per dm cube or millimole per dm cube to about, let me see over here, this is the initial rate of reaction, by the way, just over there. All right, so you can say maybe between those two values, maybe like 0.3 or something, you can say that the rate of reaction increases, and the reason for that is that the active sites are not fully occupied, and so even as you add more substrates, there are active sites for the enzymes to bind to. All right, however, after a while, beyond this value here, which is 0.03, where it starts to flatten off, 0.3 rather, where it starts to flatten off, you can then say that all the active sites have been occupied, and so even though you are adding more substrate, that doesn't result in a higher rate of reaction or doesn't increase the rate of reaction, simply because everything has been occupied. So again, I can explain this to you using, let's say, a door or a party at your school, which I think I've done before, but let's do it again. So let's say that there is a party at your school hall, and your school hall has only 2 doors. So this is door number 1. Okay, by the way, this is one door. I know it looks like creepy eyes, but that's just one door, and this is door number 2. All right, now let's assume that these over here are the active sites for the enzymes. Okay, and all of you attending the parties are the substrates, so you're going to be entering into the parties. There you are. Okay, now in this case, let's say the doors can only accept 1 person at a time. Think of them as those doors. I don't know if you've ever been to a bank where you have to walk in one at a time. You walk into a door, it closes, you hold up your hands, and then you have to walk through. So let's assume it's one of those doors. So initially, when 1 person arrives, it's very easy. So you guys, these are the substrates, these are the enzymes. 1 person arrives, or even 2 people arrive, it's still easy because 1 person goes in very quickly, and then the reaction just continues like that. Okay, and it's also happening over here. However, what will happen if there are maybe 20 of you arriving at the same time? The doors would not be able to let you in as quickly as they would if you were arriving maybe 1 by 1 or even 2 by 2, right? It would not be the same speed. So if there were 20 of you arriving, even though you're increasing in concentration, you're the substrate, but the active sites of the enzyme are occupied throughout. And that is why at the initial rate of reaction, when you're increasing the substrate concentration very slowly, the enzyme is still functioning very well because there is a lot of room for it to still take up substrates. However, it gets to a certain point where the concentration of the substrate is so high that the enzyme is not able to boost the reaction rate any further. So the reaction rate flattens because the enzyme active sites have been saturated. So that typically is the explanation that you need to give here for 3 marks. And I know I didn't put in exact words, but I hope that you can do that and just answer the question. Okay, let's look even further. Here it says the students determined the Km for radish peroxidase as 0.1 millimole per dm cube with reference to figure 4.1. Describe how they determined Km. So in order for you to determine Km, what you can do in this case, even though they don't really give you any clear instructions regarding this, I always tell students that the best thing to do is to go to the graph and draw a line. This needs to be a straight line, by the way, so please don't draw what I have. Don't make the mistake I just made. Draw a line where the rate of the reaction flattens off, okay, which is somewhere over there. So this would be what we call your Vmax. All right. And then your Vmax here is sitting at around, let's just say maybe 5.5-ish or thereabout. So we can actually just go down, or we can even say 6, I don't know. But what you're basically trying to do is you're calculating your Vmax. I'm just trying to see if my drawing is right. Yes, it is. So let's assume that's 5.5. If you divide 5.5 by 2, I think you get 2.75. So you can maybe come somewhere here, and this would be your half Vmax. Okay, so this is half Vmax. Now Km is the corresponding value on the x-axis for half Vmax. So you simply draw a line there and then you trace it down to the bottom. Okay, that's obviously not a straight line, but I think if I draw it properly, it would actually come down to 0.1 there. There we go. Okay, well, anyway, the point is it's around the region, so you can just deduce. It's not like they asked you to draw this, but I'm saying that is how you determine it. So what you can do in this case is that you can say the student first determined Vmax. You only have to write 2 points. Students first determined Vmax and use that to calculate the value for half Vmax, and then the students extrapolated from the graph the corresponding x-axis value of half Vmax, and that would give you full marks. Okay, let's look at the last one. It says a further investigation found that the Km for carbonic anhydrase, so carbonic anhydrase is a whole different enzyme from what we've been dealing with, is 12 mole per dm cube. And then here it says describe the role of carbonic anhydrase in the transport of carbon dioxide. So carbonic anhydrase is an enzyme that you will encounter in chapter 8. So if you haven't watched the videos on transport in animals, please go and do so, because these questions are important. And also I think what should come out clearly to you is how in paper 2 there is a merging of topics under one question. So you started off thinking you were only doing stuff about enzymes, which you are, but now it's taking you to think about question 8, which also has enzymes, but not within the context of enzymes as explained in chapter 3. So make sure you study all your chapters. Please do not play any more when you're studying. Don't be like, oh, my spirit is telling me that they will not bring out chapter 2. Don't do that. Please don't do that. Read every single chapter to prepare. Okay, so what is the role of carbonic anhydrase in the transport of carbon dioxide? First things first, carbonic anhydrase is the one that catalyzes the reaction between H2O and CO2 in the body. Okay, and that's one of the ways in which we transport, and I think from there we form bicarbonate, if I remember correctly. I think we form that, and we also form some hydrogen ions right there. So it forms carbonic acid and hydrogen ions, and then you can then say that the carbonic acid would diffuse into the plasma of the membrane, and you can also speak about how it helps to remove the large quantities of carbon dioxide from the body. Okay, and also another thing that carbonic anhydrase does, because this is a reversible reaction, is that it converts this back to CO2 at the appropriate time in order to transport it out of the body as a gas, but that depends. But again, this is a chapter 8 question, so make sure you read all chapters. Let's look at chapter 5. Much success was made in reducing the number of cases of malaria between 2000 and 2015, so within 15-16 years thereabout. Explain how malaria is transmitted. So again, in this case, please always mention, whenever you mention the female Anopheles mosquito, that the mosquito is a vector, right? Please mention that the mosquito is a vector. Don't mention that the mosquito is the one that causes malaria, because malaria is not caused by the female Anopheles mosquito. Malaria is caused by Plasmodium, right? So please always bear that in mind. Okay, malaria is caused by Plasmodium. How malaria is transmitted: the female Anopheles mosquito takes blood from an infected person and then inserts that into an uninfected person when it's taking up another blood meal, and as a result of that, the infective stages of Plasmodium would be transmitted into the uninfected person, leading to malaria. Again, please just write, don't write beyond the space that's provided, so don't write an entire essay. I always tell students this. I have students who sometimes even go to the extent of drawing their own additional lines. That is not necessary. That already indicates that you're not really clear on what the question is asking you, so you're trying to fill up space as much as possible with the hope that the person marking the paper will somehow, find the right answer in all your gibberish. Please don't do that. Right, be clear on your answers and be very precise. Oh, okay, I thought there was more on that slide. For some reason there isn't. Let's look at this. Diagnostic test strips for malaria contain monoclonal antibodies. The test strips detect antigens produced by the pathogens that cause malaria. Figure 5.1 shows stages in the production of monoclonal antibodies. The information in three of these stages is incomplete. So monoclonal antibodies, I think that you come across that when you do, I think it's either immunity or infectious diseases. I'm not really sure, but it's one of the two. All right, so please make sure you study those chapters really well. Let's see what happens here. So malarial antigen injected into mouse, immune response occurs in the mouse, and the mouse will then make specific B cells. So over here already this is quite easy because they tell you specific dash divides to form many plasma cells. Which immune cells form plasma cells? They are the B cells, or if you're feeling fancy you can say the B lymphocytes. Okay, the B lymphocytes are the one responsible for the formation of plasma cells. All right, then here plasma cells are extracted from the spleen of the mouse. Then you have the myeloma cells that are also added, and then the plasma cells will fuse. That is why they are called hybridomers. Well, you make hybridomers at the end. So they fuse with the myeloma cells to make hybridoma cells. I don't want to go into too much detail here, but I have explained this on the immunity. So please go and watch chapter 11 videos to get an idea of this. Please, please, please do so. And I know I'm saying please a lot, and some of you are probably like, oh, well, I'm not really bothered. I can remember this stuff. But I have seen students freeze a lot in the exams because some of these terms just won't register with them when they get into the exam. So it's always a good idea to refresh your memory by watching, okay? Then let's look at C. So these are two diagnostic test strips for malaria. So over here, CTSA, this is a negative result. That's one, so it shows 1 bar when it's negative, and it shows 2 bars when it is positive. Whenever you're giving something like this, please pay attention to the graph, to the image, and just see what you can deduce before you even go on. A sample of blood from a person suspected of having malaria is put into the well labeled S. So you put the blood here. Okay, this is our starting point. A buffer solution is put into the well labeled A. So because it's a buffer solution, let's use a blue color. So there we put our buffer solution. The buffer solution moves the blood towards the result window. So we can use a black arrow here. So it means the buffer solution starts to flow in this direction and it moves to the blood window. Okay, at position C, a line at position C indicates that the test is working properly. A line at position T indicates a positive result. So it means that as this buffer moves this blood over here, if there's a line here, as you can see in this section, that means it's a positive result. This just shows like it's a control. So I'm guessing that's why they label it C. So C would most likely stand for control, T would most likely be tests, S would be sample, and A would probably be, I don't know, just thinking out loud. State 3 advantages of using test strips for malaria as shown in figure 2.2. So first things first, your results would be accurate. Well, based on the fact that you have your monoclonal antibodies on there. So the results would be accurate. The results would also be quick. Obviously it's hard to tell if these things are cheap, or because it doesn't really tell us if they're expensive or not, but I guess you can say that the results would be cheap. Something else that I'm thinking of is that if these test strips were widely distributed, many people would not self-medicate for malaria without actually confirming they have malaria. So if you live in countries where malaria is endemic, you've probably noticed that people there, if they feel a little bit sick or they have a bitter taste in their mouth, the conclusion is usually, oh, I've come down with malaria, so I need to get treated. And so for that reason, they start to self-medicate, even though it might not be malaria. So I guess having test strips like this would help them with that as well, so that they don't have to just self-medicate indiscriminately. Okay, let's look here. Then it says that the highest number of cases of malaria occur in sub-Saharan Africa and Southeast Asia. Discuss the factors that determine the global distribution of malaria. So here you have to give 5 different points, and I know for some students that might be difficult. But first thing I want you to know, if you're a student living in sub-Saharan Africa, this should be quite easy. Not all countries in sub-Saharan Africa have malaria, just by the way. Some countries don't. Like some parts of South Africa, malaria is unheard of, and if you were to indicate that you had malaria, then you might be in quarantine, which I guess is not really new now that we are living through the time of COVID. But first things first is that you can say here the factors that determine global distribution is that the regions where malaria is endemic are regions that are very hot and humid. All right, so those are places where malaria thrives. They are hot and humid. You can also say where the female Anopheles mosquito thrives. So where it's able to survive, that would also determine. So the fact that it's not in other regions of the world just shows that the climate there does not support the proliferation of the female Anopheles mosquito. Then you can also speak about the fact that the rainfall also helps. So most of the places where you have malaria, you also have a lot of rainfall, and that's because they have a place where they need to settle their eggs on the surface of water in order to help them grow more or increase in number, if you can call it that. So you can say that also you can also mention that maybe the control measures are poor in those regions. Again, because malaria is endemic in some of these regions, people don't really make a big deal of controlling it because the immune system has learned how to live with it, and so for that reason people don't necessarily bother much in that regard. Also, you can also speak about the fact that people migrate within these regions. So infected people might migrate to somewhere else, and as a result of that might be transmitting malaria. But I am a little bit hesitant to put that as an answer, because at the end of the day you might be carrying infective stages of plasmodium, but if there is no vector to transmit that plasmodium from your body, there is no way for you to transmit it to other people, or that would be the thinking that I have. But in this regard, just think about the social and environmental factors that, and also the climatic factors, which are environmental factors that might affect the distribution of malaria or any other disease for that matter. Because whenever you get this type of questions on infectious diseases, they tend to ask you this. We are now on the last question. This again will be shorter than the one for paper one. Let's look at this. So it says here that lysosomes are cell structures that contain enzymes known as acid hydrolysis. Figure 6.1 shows some processes that occur in animal cells. Okay, let's look at this. So over here is a bacterium. First things first, whenever you're looking at this, just look at the arrow where the arrows are pointing for different things, okay? So you can see that there's an arrow pointing inwards here, but over here there are arrows pointing upwards. So just bear that in mind. So it says name the structures labeled A and E. Let's see, where's A? So this is A. A is certainly the rough endoplasmic reticulum. And why do you know that it's the rough endoplasmic reticulum? Because it has all these tiny things sitting on it over here. All right, that's the rough endoplasmic reticulum. Something else I also want to point out here is that many students struggle with differentiating between the endoplasmic reticulum and the Golgi body. Please look at the Golgi body and notice that there are no, I'm going to use a red marker. It's not a continuous structure. Can you see that? It's like they're singular worms, if you want to call them that. Whereas with the endoplasmic reticulum, it's more like a continuous maze of some sort. That is how you tell the difference between the two. So A is the rough endoplasmic reticulum and E is the Golgi body. Then it says here, state the function of the structures labeled F. So let's see, where is F? So F is sitting over here. And if we're looking at this, this is also F, by the way, just because it's not labeled, you can just sort of deduce that it's those types. So here, these are the rough endoplasmic, this is the rough endoplasmic reticulum, and it's releasing these little things and taking them to the Golgi body. What that tells you is that we are dealing with vesicles. Okay, so in this case, you can say that it is transporting molecules from the rough endoplasmic reticulum up to the Golgi body. All right, and that would give you a full mark. And then name the process by which bacteria are taken into the cell at C. So that process definitely has to be phagocytosis or endocytosis, because again, pay attention to the arrows. So in this case, the bacteria are not leaving the cell, they are coming into the cell, and then they are swallowed into these vesicles. All right, so if they're being swallowed into the vesicles, it means that they're being taken in. That is endocytosis. For those of you who might not know, and there are two types of endocytosis. I think there's phagocytosis, which is your uptake of solid material, and you have pinocytosis, which is the uptake of liquid material. All right, just make sure you check on that. Okay, let's look at the last two questions. With reference to the processes occurring at B and D in figure 6.1, outline the role of acid hydrolysis in lysosomes. So let's just look at B and D. So here's B over here and there's D. So with D, we can see that it's the digestion of bacteria. And with B, I think these look like old plant cells. I'm just saying that because I can see this looks like a cell wall and maybe like a cell membrane on the inside for all of these cells, and then when they get to B, they start to look a bit different. So I think one thing that I would say here is, first of all, they aid in the digestion of pathogens as shown in D. So when a pathogen invades the body, the lysosomes will swallow it in the process of phagocytosis and digest it so that it doesn't cause any harm. But what they might also be doing is that they might also be helping to break down old cells and cell organelles that are no longer functional, and so they break those down, and obviously by breaking them down they might release some components into the cell environment that can be reused, because sometimes a cell that's old doesn't necessarily mean that every little part of it is no longer functional, so they might be able to do that as well. Carrier proteins in the membranes. Oh, and also something I would say, just by the way, is that because they are hydrolysis, that they help in hydrolytic reactions, so they help with breakdown through hydrolytic reactions. So I think that would also be helpful, just based on the name of the enzymes. Then the last question, carrier proteins in the membranes of lysosomes maintain a lower pH than the surrounding cytoplasm by moving hydrogen ions. Suggest how the carrier proteins maintain the lower pH within the lysosomes. So in this case, I think something that you would say is that the carrier proteins in the membranes of the lysosomes, for them to move hydrogen ions, first of all, you need to know that they have a proton pump, okay? So you can then speak about how they pump protons into the lysosome. And as a result of pumping the protons into the lysosome, they are creating an acidic environment which would have a lower pH than the surrounding cytoplasm. And that should give you your full marks. But yeah, this is the end of this paper. I hope you found it helpful, and I hope that you are studying hard towards the exams. I'm going to do some more shorts videos just so you can revise concepts, but I hope that this has been a great learning experience for you. The next paper I will do will be paper 4, and for those of you who are writing, I want you to know, based on the trend I have seen in the past for the exams, paper 4 is usually your first exam. So you should be aware of your timetable by now, but please make sure you start studying very hard for that one. All right then, until the next video, goodbye.